morning. Let's see, Bryant is here. Curry is online. Darby is here. Fisher. Hamilton. Hellman. Ashley is here. Crystal is here. Taylor is online. Norris is here. Rollins is here. Saunders is online. Shamblin is here. Vargas. Did I miss anybody? Give it a little bit of time. It's not quite it yet. But now it is. Okay, so announcements, since we're recording. Fisher is online. 1 second. There we go. Actually, I guess before I do announcements, let's do the first attendance word. So again, as a reminder, if you're watching this live, watching the live stream, then you need to send the words in after the lecture. And the first word will be agent. And as usual, well, that has been the case recently, use that word in a sentence when you send it in. Anyway, let's see. Announcements. I'll do less important ones first. So this will be the one last day where I'm doing office hours 100% online. So I'll be online today for office hours if you need me, not in person. Speaking of which, sort of kind of, the exam will probably be this week. At this rate, we should be able to finish this chapter today or on Wednesday. Therefore, the exam will be on Friday at this rate. I should have to be prepared for that. You need to be here in person for those of you who are usually online. Bring your computers. It's again about 50 questions. You have 50 minutes to take it. So if you want more time to do the exam, you need to contact me today and say, hey, can we find a different time to meet online so I can proctor the exam and you can have more time. But you need to let me know today. Last time I had somebody tell me like the day of, hey, can I take the test some other day? And I did. They did, but we planned ahead this time. So any questions about and then also for the exam, we're going to cover mitosis, meiosis, right? That chapter about cellular reproduction. And the one after that, which was genetics, and then finally this one, right, where we're talking about DNA to RNA to protein and DNA replication. So it's all about basically cellular reproduction is what the exam's going to be about. I highly recommend doing the study guides. That's what I use to write the exams. And if you want, you can meet me, find time to meet me online, and I will go through those study guides with you and say, this is the right answer, this is the wrong answer, this is why this is right, this is why this is wrong. This definitely will not be on the exam, this definitely will be on the exam, this might be on the exam, all those things, right? So you have me at your disposal if you want to help prepare for the exam. And then finally, regarding exams, once we take exam 3, which would be this one, you can no longer retake exam 2. So if you haven't retaken exam 2, I recommend it. Because again, even if you do worse, we'll just keep your high grade, so you have nothing to lose. Any questions about that? All right, lab will be a lot like last week. It's another in-person, we're going to meet, but it's another online lab, right? So we're not going to physically be doing any experiments, so bring your computer to lab. And if you get it done before Wednesday, then you'll have to come to lab. That's it. Does anybody have any questions? All right, let's jump back into it. So remember, we were talking about this right here. We're going from DNA to RNA to protein. Again, you can study however you want, but my recommendation is to memorize that first. Memorize that that's the process. That's the flow, right? Memorize the fact that we go from DNA to RNA to protein. Then once you have that memorized, then memorize what that's called, right? So DNA to RNA, as you learned last week, is called transcription. And we're about to talk about that. And RNA to protein is called translation. And that's what we're going to talk about later today. Any questions? All right, let's jump into it. Remember, we did the overviews. Now we're going to get more specific here. Transcription. This is the transfer of genetic information from DNA to RNA. And of course, I'm not going to ask you the definition of transcription. I'm going to ask you what happens during transcription. Or I might say, hey, this thing's happening. Was that transcription or was that translation? Anyway, when this happens, the DNA strand separates where the process begins. I'm not necessarily going to ask you that. So I'll put a little X through that. When this happens, when that DNA strand opens, and this is gonna be a horrible picture, but imagine, it looks like that. So there's your bases hanging off the DNA strand. And once it opens up, only one of these strands is used as a template. The other strand is basically just useless as far as we're concerned. I'm not gonna ask you that either. What happens is, and I'll show you a better picture later, when this opens up, the RNA polymerase, it reads the code, it reads the base, right? So let's say, for example, that's an A, maybe that's a T, maybe that's a C. The RNA polymerase reads it and brings in the complementary code, right? So if it's reading this C on the DNA, does anybody remember where it goes to C? Because you need to know this for the example. Good. If it reads a C, it's going to bring in a G nucleotide for an RNA, right? And this one's T, so it's going to bring in an A. This one's A, so it's going to bring in a U, right? Normally it'd be a T, but we're talking about RNA, not DNA. Any questions so far? All right, like I just said, now you can see it in writing. The RNA nucleotides follow the same base pairing rules, so like I already said, like you need to know. A usually goes with T, but there is no T, right? So in this case, A goes with U, T goes with A. Again, if that's DNA, that's RNA. T goes with A, C goes with G, and you need to know that. And again, like I said in the last slide, now you can see it right. The machine that's doing this, the enzyme, is called RNA polymerase. So those are the two enzymes that are going to be on the exam for this chapter. You need to know them both. There's only two, and what they do, it's in their name, right? DNA polymerase makes DNA. RNA polymerase makes RNA. It's pretty simple. Any questions about this slide?
Hey, is the D and R the ones that go together, or did you just?
No, those are, I'm just saying, if this is the DNA basis,
Oh, okay.
then that's the RNA equivalent, right? So if it reads an A, it's going to bring in a U. If it reads a T, it'll bring in an A. If it reads a C, it'll bring in a G. And again, I keep saying this every time we bring it up. When you get the question on the exam that says, hey, here's a strand of DNA, right? So it'll be a bunch of letters. And the question will say, transcribe this. So remember, when you're transcribing something, that means you're making a strand of RNA. So any question, or excuse me, any of the options on the multiple choice exam that have a T in there, you know that's not the correct answer. You can just go ahead and eliminate it. Don't waste your time comparing it to the question. Right? If it has a T in it, it's not RNA. Therefore, that's the wrong answer if the question asks you to transcribe something. And also one last reminder too, there'll be at least one question on the exam where I give you a whole strand of DNA and I say transcribe that, and then you'll figure out the RNA. And then I'll say translate that. So then you'll take the RNA and translate that into amino acids. We talked about that last week. I'll talk about it again once we get to it. So any questions about this before I show you a picture of what's going on here? All right, this picture's not from your textbook. This is from the old textbook, but it does a great job of showing what I'm talking about, right? So the DNA strand is opened up, because those are just hydrogen bonds, right? So we just slip the bonds. RNA polymerase comes in and it reads the code. Here's a T, right? So it's bringing in an A. Here's an A, so it's bringing in a U. And it's going to make those covalent bonds eventually. Here's another A, so another U comes in. That's it. Any questions about this? All right, just to make things a little bit more difficult, transcription itself is broken down into 3 stages, and you need to know them. Initiation of transcription, RNA elongation, and termination of transcription. And I put it in bold, the words you can shorten it. You can just memorize initiation, elongation, termination. And for the most part, the names kind of explain what they do. I see you guys taking notes, so I'll wait a second before I move forward. Next word for attendance, I'll underline it. And again, if you're watching the video later today, or whenever you watch it, send that word in a sentence. Anyway, any questions about this? All right, let's talk about the first step, initiation. So the start transcribing signal is a DNA sequence called a promoter, so you do need to know that. Another way I could write that bullet point is the promoter is where transcription starts. So that would be most likely the question. Where does transcription start? And it starts at the promoter. So again, the process, first step, we call that process initiation. Physically, the place on that chromosome is called a promoter. Obviously, it's going to be at the beginning of a gene, right? You don't want to have it in the middle of the gene or the end of the gene, because the whole idea is to read the code. And obviously, that's where the RNA polymerase is going to attach. So you can read a lot about that if you want for independent work. It's actually pretty interesting, because there are, our cells do need to regulate our genes. Sometimes genes need to always be on. Sometimes they need to be very rarely on. So there needs to be basically on and off switches. It's pretty complicated, but interesting. Anyway, so that just means initiation is basically just when the RNA polymerase attaches to the promoter. And that's it, right? That's the beginning. Initiation is just that, the machine, if you will, the RNA polymerase attaching to the chromosome, attaching to the DNA at the right spot. And the right spot is called the promoter. Any questions? Right. So, the second phase of transcription is called elongation. Like I said, you need to know that. And it is exactly what it sounds like. Wow. Is that one of you guys, or? Oh, it's somebody online? Okay. I was wondering, that's kind of odd. 1 second. See if it's not muted here. There we go. They're muted now. For their own sake. Could be something embarrassing. But anyway, back to it. Again, the second phase is called elongation. It's in the name. What's getting elongated? Well, the thing that we're producing, which is the mRNA. Elongation is simply the mRNA getting longer. As that's happening, as you can see in this picture that's not from your textbook, the RNA kind of peels away from the DNA. It goes in its own direction. We'll talk about what happens to that later. Also, I'm going to ask you, but the DNA, as you might imagine, it comes back together, right? Like we have to open it up to read the code. But once we're done reading the code, it comes back together. Pretty simple stuff, right? Any questions about that? All right, the last part, again, very easy. It's in the name. Termination, right? We're terminating, we're ending it. If you've been terminated from your job, your employment has been ended, right? Termination means end. I guess the only difficult part to this is knowing where it happens, and that is called the terminator. So the whole process starts at the promoter and ends at the terminator. And as you might imagine, it's kind of the opposite of the first phase, right? So now RNA polymerase detaches. It goes its own way. The DNA comes back together. The mRNA goes on and does its own thing, which we're about to talk about. So anyway, any questions about the 3 stages of transcription? All right, let's move forward then. Here's a picture of everything we just said, all in one big picture. This is not from your textbook. This is from the previous textbook. It's hard to see, but it's a blue strand of DNA, a little green strand right there, a little red strand right there. Of course, it's not like that in nature. We're just trying to visualize things. So that's the promoter. This is where the RNA polymerase attaches. It goes down, right? It goes down the RNA, or excuse me, the DNA reading the code. That makes a strand of mRNA, or at this point, it's just RNA. And then at the end, it detaches, everything goes its own way. Pretty simple. If you download the PowerPoint, you can watch this little animation that shows you what's happening. So that's it for transcription. Next will be translation, but there is one little in-between spot. First, this RNA has to be processed. Your book does point out, and I'm not gonna test you on this, I'm not gonna ask you, but in prokaryotes, remember there's no nucleus in prokaryotes, they're a lot more simple. The RNA is transcribed right away. So as the RNA polymerase reads the DNA in the bacteria, right away, that RNA is then translated into protein. But for us, for eukaryotes, we're more, I won't say special, but more complicated, right? So our RNA needs to be processed before it can go out into the cell. So that's what we're about to talk about. Here we go. So you need to know this happens. Cap and tail just basically means it's exactly what it sounds like. We've got some enzymes that put a cap on there, and some enzymes that put a tail on there. And yes, technically that tail is a bunch of A's. You don't need to know that. Just know that there's a cap and a tail. And your book does specify which is which. As far as I'm concerned for the exam, you can just think of them as the same thing, cap and tail. Same process. Because mostly they protect it, right? Protect the mRNA, because your cells like to destroy things that don't belong in there. So this is a way of letting your cells know that that RNA does in fact belong in the cell. Because as you're going to learn later, but basically viruses, all they do is put in RNA or they put in DNA. And obviously your cells need to fight against viruses, so they destroy DNA, they destroy RNA. Unless, again, it has the cap and tail. And also, as you're going to learn later, it also helps, there's a thing called a ribosome. And again, I'll teach you about that later. That's the thing that does the translation. It's the thing that reads the code and makes the polypeptide, the protein. So again, it's cap and tail, helps those ribosomes recognize it as a gene, as a RNA that's ready to be translated. So again, when you're reading about this in your book, it goes into more detail and then distinguishes between the differences between the cap and the tail. As far as I'm concerned, it's all the same. Any questions? All right, another thing is RNA splicing. Here's where it gets a little bit more complicated. So we have different regions of the DNA, right? So if this is the RNA that came from the DNA, I guess technically it's pre-mRNA. There's some spots of the RNA and we receive the DNA that just aren't read. Those are the introns, the in-between parts. The exons are the parts that are actually expressed. Those are the codes that actually say, all right, bring me this amino acid, bring me that amino acid. So you do need to know the difference between exons and introns. And the way I always remembered it as a student was the exons are expressed. Right? Those are the ones that are getting translated and turned into proteins. And the introns are just the in-between stuff. That's how I always remembered it. So any questions about this? See note taking, I'll slow down a little bit. So again, yes, you need to know that RNA splicing happens in between transcription and translation. You need to know that we leave the exons in and get rid of the introns. The introns sometimes it's called junk DNA, but if you were to look deeper into it, it's not necessarily junk. It has purposes that we just don't discuss in this exam or in this course. Oh, and also, by the way, both of these, the RNA splicing, also the cap and tail that we discussed previously, all of this is happening inside the nucleus. This is all preparing that RNA to leave the nucleus, right? It's not ready yet. It has to do all these things before it can leave the nucleus. Another thing about the RNA splicing is that, well, that's it. We already talked about that, but there is something called alternative splicing, which makes sense. Not that you need to know this, but think about this. Humans have about 21,000 genes. And remember, genes are instructions on how to build a protein or a polypeptide. In this chapter, those words are interchangeable. So how is it that we have 21,000 genes, but we have thousands more proteins or polypeptides? Any guesses? Because when you think about it, on the surface, that does not make sense. Genes are instructions on how to build proteins. So where are we getting these other proteins from if we only have 21,000 genes? So no guesses? All right, come back to it. I'll come back to that too. Alternative splicing, that's how it happens. So again, here's exon 1, exon 2, exon 3, exon 4, exon 5. This picture's not from your textbook. In this case, again, with splicing, we get rid of the introns, right? So we get rid of this portion, we get rid of this portion, we get rid of this portion, we get rid of this portion. And then you're left with an mRNA that has exon 1, exon 2, exon 3, exon 4, exon 5, right? And when you translate it, you get that protein. However, there's alternative splicing. So in this case, we have exon 1, exon 2, we skipped exon 3, but we have exon 4 and we have exon 5. So when that's translated, you get this protein. And you can see they're similar, but they're different. And of course, we have this one, so exon 1's there, exon 2, exon 3. This one leaves out exon 4 and goes directly to exon 5. When that's translated, you get that different protein. So here we have, and again, this is an oversimplified example, but here we have one gene that was spliced 3 different ways, therefore we get 3 different proteins, all from one gene. Any questions about that? So that's it for transcription. Now, once this happens, right, we have the cap and the tail, we've done the splicing. I'll back it up to the other picture. Right, now it's ready to leave the DNA, right, or excuse me, the nucleus. The DNA is ready to leave the nucleus, it's ready to go out into the cytoplasm, into the cell, where it can be translated. And that's what we're going to talk about next. And I'm going to teach this again the way your old textbook did it, because before it talks about the process of translation, first they introduce you to the parts. Right? So first we'll talk about the parts that are involved, and then we'll talk about the actual process itself. You need to know this. You need to know the ingredients, so to speak. The first important ingredient of translation, obviously, is the mRNA. That should be obvious because that's what we've been talking about, right? Transcription was the process that made the mRNA. And then we did the cap and the tail, did some splicing. Now we have our mRNA. It leaves the nucleus. It's ready to be translated. Then there's the machine that does the translating. And I already told you what that is, and I'll give you more details later, but it's the ribosome. And of course, all this requires enzymes and energy. Usually that energy is ATP. There are other sources of energy. You can look it up if you want for independent work. And then translation requires 2 other very important components. Actually, I need to fix this slide because the machine that does the translating is actually ribosome. So those are kind of saying the same thing. Anyway, so yeah, we have the ribosome, which does the thing. That's the enzyme, so to speak, that does the actual translating. And then a different kind of RNA called tRNA. So there you go. Now you've been introduced to 2 different types of RNA, and you need to know the difference between them. mRNA has the message, right? It's the messenger RNA. That's what the M stands for. tRNA transfers things. You'll learn what they transfer later. But yeah, you'll need to know the difference. And then later, I'm going to give you yet one more type of RNA. So for this exam, you're going to need to know the 3 different types of RNA. There are many more types, but we just don't discuss them in this course. So any questions about the slide? All right. Here's the next word for attendance. I'll circle it. So again, you're all 9. Email me those words as soon as class is done so you can get your points. If you're in person, you can email me for extra points. If you're watching this video later, send me a sentence using that word. All right. So no questions. We will move forward. Again, right, we're talking about translation, right? So just like any other kind of translation, even if this was in a non-biological sense, we talk about translating from Spanish to English, you need an interpreter, right? Somebody who does the translation. Same here. So we need something that can convert the 3-letter nucleic acid words, which you already know as codons, because you learned that last week, into amino acid words, right? Because again, we're translating. We're going from one language to the other. We talked about this on Friday. Transcription, we're just going from DNA to RNA, right? That's the same language. That's the nucleic acid language. We're just copying the message. Now, we need to take that message and translate it into something 3-dimensional, right? We're making a polypeptide. So finally, we get back to what this does. The transfer RNA matches the amino acids to their codons, and that's what forms the polypeptide. Another way of writing this, and if you're taking notes, you could write this down. A better way of writing this, I think, would be transfer RNA transfers amino acids. That's what it's transferring. Because remember, from way back at the beginning of the semester, proteins are just a bunch of amino acids put together, right? 20 different amino acids, we put them together, just a long chain of amino acids, and then they fold into the correct shape, and that's what makes it a protein. So, obviously, we need something to bring the amino acids in, and that's what the tRNA does. So it picks up the correct amino acid, then it brings it, and it reads, it's the thing, it actually reads the mRNA and says, okay, this must mean that I need this particular amino acid. And not that I'm going to ask you this, but as you might imagine, because it's doing this, that means there's one tRNA for each amino acid. It is a particular tRNA, and its job is to carry that one amino acid. And I'll show you a picture of one on the next slide. I don't have any questions so far. So again, the two different types of RNA that you're familiar with now are the mRNA, which is the message, and the tRNA, the transfer RNA, which is transferring amino acids. And we'll talk about it a little bit more here. I'm not gonna, let me see. Yeah, I'm probably not gonna ask you any of this, but just so you know, this is how it works. It's made up of a single strand of RNA. I know that looks like double-stranded, right? Because we have hydrogen bonds, just like we would between two strands of DNA. But what's actually happens is one strand of RNA, and it's kind of folded up on itself. Not that I'm going to ask you that. But yeah, it twists and folds. It has to have the correct shape. It forms double-stranded regions, like I said right here. But again, I'm not going to ask you any of this stuff. Here's the more important part. At one end of the tRNA is something called an anticodon. So that's the thing that's reading the mRNA. It reads the codon and says, all right, that's my job. That's what I'm here for. That's my code. So now I need to bring in my amino acid, because remember, there's one particular tRNA for each amino acid. And the next picture I show you is really going to clarify this. So yeah, on one end of the tRNA, you have the anticodon. That's the thing that's doing the translating, so to speak. That's the thing that's reading the message. And on the other end, obviously, since it's a transfer RNA, it's transferring amino acids, that's what's on the other end, right? So you have the amino acid on one end, you have the anticodon on the other. So, any questions about that? Or I showed a picture, basically the words that are on the slide. All right, here's the picture. Not from your textbook. So this is what it actually looks like, right? Well, it's more accurate than what's on the right. But the one on the right's better for us. This suffices, right? This is a lot easier to understand. Like I said, on one end, we have where the amino acid attaches. On the other end, you have the anticodon. That's the thing that's reading the codon on the mRNA and says, okay, that's my code. Any questions? All right, so that's tRNA. Let's talk about the other important part, which is ribosomes. Ribosomes are organelles, so they don't have them in prokaryotes, only eukaryotes have them. And basically, I like to call them the machines. These are the machines that are doing the translating because they coordinate the functioning of the mRNA and the tRNA. So remember, the mRNA has the message, right? It has those A's and A's and U's and C's and G's, those three-letter words, and each one of those words says, hey, bring me this amino acid, bring me that amino acid, right? That's what the codons do. That's what they say. The tRNA reads that codon and says, okay, you want this amino acid? That's me, and it brings it. Now the ribosomes put all this stuff together. So it's not just floating around happening in your cell magically, right? The ribosomes bring it all together. Technically, we say then that the ribosomes are the things that make up the polypeptides. What I'm not going to ask you about, but I will talk about it in a little bit of detail moving forward. Technically, ribosomes have two subunits. There's a large subunit and a small subunit. Each one of those subunits is made up of proteins and yet another type of RNA. I guess I made that X too big. You do need to know the third, like I said, the third type of RNA that you need to know about is ribosomal RNA. That's it. Now you've been introduced to all three types of RNA for the exam. mRNA, which carries the message. tRNA, which carries the amino acid. And rRNA, which is just what ribosomes are made of. I might not even have any questions about rRNA, because really, ribosomes are important, but it doesn't matter to you, right, what they're made of. They make proteins. They are made of proteins. They're made of RNA, but for you, it doesn't matter. Now, the ribosomes, and you'll see this more when I show you the picture, they have a binding site. Obviously, if they're bringing all this stuff together, then they need a site for it, right? So they have a binding site for the mRNA, and they have a binding site for the tRNA. And your book does get specific. It says that the binding site for the mRNA is on the small part of the ribosome. The binding site for the tRNA is on the large part, but you don't need to know that. So any questions about this slide before I show you basically a picture of what's on these words? All right, this picture is also not from your textbook. Actually, I need to look at your textbook. Usually, in a 101 class, you're just going to look like this. You're going to have the A site and the P site, and I'll explain what those are later. But in all actuality, there's a third site. And for some reason, in 101 classes, they never talk about that third site. I mean, I know we're trying to make it easier, but anyway, the only reason I'm telling you that is because I don't remember what your textbook has. So if you're reading and studying your textbook and it talks about a third site, I don't want you to get confused. It's just because the last textbook and most textbooks do oversimplify it and leave out the third site. Anyway, like we've already said, the ribosome is made up of a large subunit, a small subunit. It brings all the parts together, right? So the mRNA attaches to the small subunit, and we'll talk about this in more detail later. The tRNA attaches to the large subunit, and that's where magic happens, so to speak. And again, so far, we're still just talking about the parts, right? We haven't even started talking about the process of translation yet. We're just talking about the parts. So are there any questions about the parts? They're all right there in this picture now, right? All the parts are here. We have the mRNA, we have the tRNA, we have the amino acids, and we have the ribosome. All the players are right there. So are there any questions about all the parts before we go into the process? All right, let's go into the process. Translation, the process. Here's some good news for a change. There's 3 stages of translation, and as far as words are concerned, nomenclature, it's the same 3 from transcription. Initiation, elongation, termination. So, same names. Similar processes. And obviously, as you might imagine, we're going to talk about each one of these separately. So, initiation. As you might imagine, logically, this just makes sense. I probably could have asked you this before I showed you this slide, and somebody eventually probably would have guessed it. Initiation is just bringing all the parts together. So all those things I just talked to you about, the mRNA, the tRNA, the amino acids, the ribosomes, right? They all have to come together, and that's what initiation is. And as I mentioned earlier, remember there's a cap and a tail, and that helps with the process. But when it comes to the discussion or any questions about translation, I'm not going to ask you about cap and tail. I just included that because when I talked about cap and tail, I told you it was going to help the ribosomes. Well, that's where it came back. So yes, I might ask about cap and tail in the context of transcription, right? Because that happens after transcription. That happens before the mRNA can leave the nucleus. But when I ask you questions about translation itself, I'm not going to bring up the cap and tail. Anyway, it also brings the first tRNA, obviously with its amino acid, and of course the subunits and the ribosomes. So all the parts are put together. That's what initiation is. Pretty simple. Any questions about this slide? All right, here's another picture of the mRNA to remind you. This one's not from your textbook either. But again, there's your mRNA, right? That red part, that's the message, right? Those are the codes, each 1, 2, 3, right? Each 3 of those letters, that's a word. It's a codon. It says, bring me this amino acid, bring me that amino acid. So that's, well, let's talk about this a little bit more. As I already said, and like I said, I'm not going to ask you this, but the mRNA is binding to the small subunit. Then the tRNA comes in, the correct one, and it reads the code, right, with its anticodon, and it comes in and attaches to that. And then the large subunit comes in. This picture is not from your textbook. And notice, like I said, it has the third site. But I'm not gonna ask you questions about that, I'm just pointing that out so you don't get confused when you're studying this, like, wait a minute, first it had 2 sites, now it has 3, just don't worry about it. Also, actually, in a sense, you can not even worry about this whole slide. Because I'm not gonna ask you to memorize the fact that the mRNA attaches to the small subunit, and then the tRNA comes, attaches to that, and then the large subunit comes in. I'm not gonna ask you all that. For initiation, what you need to know is that's when all the parts come together, and, well, I guess you need to know this middle bullet point. Where does this start? It starts at the start codon. So remember, transcription, you had to know where it started, right? Transcription starts, initiation for transcription starts at the promoter. For translation, it starts at the start codon. It's an easy question to miss. People miss it a lot. Doesn't make me sad like some of the other questions, because I get it. That's easy to mess up. But yes, translation starts at the start codon. Transcription starts at the promoter. Anyway, any questions about initiation? Again, it just brings all the parts together, starts at the start codon. Here's another picture, not from your textbook. This is from your previous textbook. But again, it's all the parts, right? mRNA attaches to the small subunit. tRNA comes in with that, reads it, recognizes or has its amino acid. And then once all that's together, then the large subunit comes in. There you go. If you download the PowerPoint, you can watch this little video, this little animation that shows it happening. The amino acids, excuse me, now step 2. Step 2 is elongation. And again, this is in the name, right? When we were talking about transcription, that was pretty easy. Elongation, what was being elongated? Well, the RNA, that's what we were making in transcription. Here, we're making a polypeptide, right? Or a long chain of amino acids. Therefore, it makes sense that elongation is just the chain of polypeptides getting longer. That's what's being elongated. But unfortunately, for you guys, it does get a little bit more complicated. So elongation itself, which is one of the 3 stages, right? Even that is broken down into 3 steps, so let's talk about this. And I'm not sure if I'm gonna ask you these 3 steps or not. But anyway, step 1 is codon recognition, right? So what happens, as we've already mentioned when I first introduced you to tRNAs, the tRNA reads the code and says, oh, that's my codon, right? So whichever particular codon or whatever amino acid that the code is asking for, particular RNA recognizes it and it attaches. Now, yes, technically this happens in the A site and ribosomes. I'm definitely not going to ask you that. Again, I don't care about the sites. There's A sites, P sites. Remember, you don't need to know that. Just know that the first part of elongation, codon recognition, right? That tRNA recognizes that code and says, oh, that's me. Any questions about step 1? All right. Step 2 is peptide bond formation. In other words, step 2 is when it actually elongates. Step 2 is when we add the new amino acid. And technically what's happening, and I'm not going to go into this much detail in the exam, technically what's happening, as you might be able to see from this picture, is, right, it brought in this new amino acid. And technically what's happening is that strand of growing RNA, or excuse me, growing amino acids, is then attached to the new amino acid, as opposed to the new amino acid being added to the strand. It's a subtle difference, but I'm not going to ask that. And yes, again, this happens in the A site. I'm not going to ask that. At that point, it leaves the tRNA in the P site. Again, I'm not going to ask that. Here's the important thing, these 2 bullet points here, right? Step 2, that's when we're actually adding the amino acids and making the chain longer, which is the whole purpose of translation anyway. So in a sense, for this discussion, you could consider this the most important part of translation, because the whole point is to make a polypeptide, right? To add a chain of amino acids. And this is the place where it happens. In elongation, specifically step 2 of elongation. Another thing I'll point out, and I don't know why they do this, just to make things more difficult for you, just to remind you, because we haven't had this discussion in a while, a peptide bond, it's just a covalent bond, except now when we're talking about a covalent bond between amino acids, they have to call it a peptide bond. I'm not going to ask you to differentiate, I just want to point that out. So when you hear me say, or when you read about a peptide bond, just know that it's just a regular old covalent bond. It just so happens that it happens to be between amino acids. It just had to make it difficult for students. I would have just called it a covalent bond, right? We don't need to give them special names. Anyway, any questions about step 2? All right, step 3 is translocation. This is really well, I won't say really complicated, but basically all the parts are just moving around. So I'm going to put an X through that because I'm not going to ask you, because like I said, I'm not going to ask you about the P side or any of that. So really, again, in my opinion, the most important thing is step 2, right? Step 2 is where we're actually adding the amino acid, or technically adding the chain to the new amino acid. But again, I won't even get into that much detail. And then here's everything in one picture, right? So this is all of elongation, right? This thing's here. There's a particular code that says, hey, I need this diamond amino acid. This particular tRNA recognizes that code and said, oh, that's me. I'm the one with the diamond amino acid. So it comes in, right? That's step 1. Step 2, like I said, the growing chain is then added to the new amino acid. And then step 3, which we didn't really talk about, because as far as I'm concerned, it's not important for us. Then all the, this tRNA is useless now, right? Because it had the amino acid chain to it, but it gave that to that amino acid. So it leaves, and then this thing scoots over, and the whole process continues. So any questions about elongation? All right. Where do you guys think? Let's see if anyone can guess this. So remember, for transcription, we talked about initiation, elongation, termination. And transcription started at the promoter and ended at the terminator. Translation, I told you, starts at the start codon. Where do you think it ends? If it starts at the start codon, if you had to guess, where do you think it ends? Where do you think the process stops? No guess? All right, here it is. It stops at the stop codon. You need to know that. So again, remember, transcription, it stops at the terminator. For translation, it stops at the stop codon. Yes, technically, the stop codon is always a UAA or a UAG or a UGA. I'm going to put a line through that because I'm not going to require you to memorize that. It's just so you know the stop codon, that's what they are. So, even though we haven't talked about this much yet, we will a little bit later. Excuse me. If there was a mutation, right, and swapped one of those letters out for another one, right? So maybe that's supposed to be UAA. If that got switched to, I don't know, UAC, right, which is none of these, then there would be no stop codon, right? And then the process would keep going and going and going. So if you had a mutation that changed the stop codon to not a stop codon, that would be the result. You have a really huge protein. Likewise, if you had some other code that used to say, I don't know, I'll use the same example. Maybe you had a code that's supposed to say UAC, and I don't have that memorized, like what amino acid that's supposed to be for. But if you had a mutation where it changed that C to an A, then you would have an extremely short protein, right? Because the UAA is a stop codon. So if you had a mutation that changed one of your other codes into a stop codon, you would have a very small protein. And remember, it's all about the shape, right? So obviously, if it's too long or too short, it's probably not going to work. Anyway, at this point, again, what you need to know for the exam, termination happens at the stop codon. And as you might imagine, it's basically the opposite of initiation. So initiation, all the parts came together at the start codon. Termination, all the parts go their own way at the stop codon. And again, this picture is not from your textbook. So again, you can see this particular picture shows 3 different sites. You don't need to worry about that. Another thing, and I don't remember if your textbook talks about it or not, this whole process, right, it goes from 3' to 5'. That's another thing you don't need to worry about. So when you're reading that and it seems overwhelming when you're studying from the textbook, don't worry about that. Just remember, I'm not going to ask you what direction it goes. You just need to know all the parts come together, and this is what happens. And then at the end, at the stop codon, all the parts go away. So any questions about that? All right, let's do an overview of it very quickly. Again, the flow of information goes from DNA to RNA to protein. You do need to know that. And again, everybody learns differently, but my best recommendation is to memorize that stuff first. Memorize the big picture stuff, then start adding the pictures, or the details. Someone will miss this one, and that one actually will make you sad. Just like people miss the questions about photosynthesis. And I purposely did one stuff that wasn't photosynthesis, and people chose that. Broke my heart. Anyway, yes, memorize that first. DNA to RNA to protein. And then once you have that memorized, memorize the fact that going from DNA to RNA is called transcription. And then memorize the fact going from RNA to protein is called translation. Then you can memorize the fact that transcription happens in the nucleus, right? And then mRNA is processed before it leaves the nucleus, and then that's where translation happens. One thing I'm definitely not going to ask you about is this. One ribosome makes an average sized polypeptide in less than 1 minute. But I'm not going to ask you that. It's a good independent work topic, though. Average size polypeptide. What is an average size polypeptide? How big is your average protein? And again, like I said, I'm not going to ask you this in this exam. For this exam, protein and polypeptide, those words are interchangeable for this exam. But I want to remind you, like we talked about way back in chapter 3, a polypeptide is nothing but a bunch of amino acids put together. It's not technically a protein until it folds up on itself and it gets its particular three-dimensional shape. So any questions about this slide? All right, here's a picture of everything that's on this slide. There's your DNA in the nucleus, right? The RNA polymerase attaches to it, a promoter. It opens it up so it can read the code. It elongates, right? It's making this strand of RNA. Then that RNA, before it can leave the nucleus, has to be processed, right? So we give it a cap and a tail to protect it and to help it be recognized by the ribosomes later. We cut out the introns. Now it's ready to go. It leaves the nucleus. Then we have, well, your book doesn't even talk too much about it. You don't have to worry about this, but obviously what we did talk about with the tRNA is obviously something has to put these things together, right? So we have the tRNA, we have its amino acid, something has to put it together. We don't talk about what, you can look it up if you want. What enzyme is it that does that? But I'm not worried about it. You don't need to worry about it either. All the parts come together in translation, right? So step 1, all the parts come together. The mRNA attaches to the small subunit. The tRNA recognizes the start codon and it attaches there. Then the large subunit comes in, and that's initiation, right? All the parts are together. Then one by one, we keep adding amino acids to the chain. The chain gets longer and longer. That's the elongation. And then finally, it hits the stop codon, and the whole process goes over, and everything goes its own way. And that's it. As far as we're concerned, that's it. Now, when you read chapter 4, I think it was, from way back when, your book actually goes into a little bit more detail about what happens, because really, it's not over. It's over as far as the exam is concerned. But more stuff actually happens. Those proteins actually have to be processed, because they don't just sit there where they're made. They have to be shipped out, right? Sometimes those proteins need to leave the cell. Sometimes they need to go to a particular place in the cell. And your cells do all that stuff, and we don't talk about it. Anyway, transcription and translation, again, that is how genes control the structures and activities of the cells. Because again, protein part, the structure, that makes sense. That's easy to think of. Like, all right, it's protein, that's the structure. But remember, almost everything that happens in the cells happens because of enzymes, right? And enzymes are proteins. So that's how it controls the activity of your cells as well. So again, this slide is just a reminder of everything we've already said. The information originates with a specific sequence of nucleotides in a DNA gene, right? So all the A's, the T's, the C's, and the G's, that's the DNA language. It's nucleotides. Just like, you know, in the English language, we have A through Z. This language, you have A, T, C, and G. Each word is 3 letters long. The gene dictates the transcription of the complementary sequence of nucleotides in the mRNA. That mRNA sequence then specifies a particular amino acid that's going to be added to that chain. And then finally, the proteins from the polypeptides determine the appearance and capabilities of the cell and organism. So that's everything we just said in that one slide. The next word for attendance will be organism. And again, if you're watching the video, not the live stream, but if you're watching the video later, don't just send me the word, just send me the sentence that's using it. Anyway, back to the review. The DNA to RNA protein pathway was believed to be the only way that genetic information controls traits. However, I should fix this. This is old. There's other stuff. RNA does other stuff that we don't even talk about in this class, and you can look it up if you want. It's in your textbook. I have to look to see what chapter it is in your textbook. You're not required to know about it, and that's a perfect place to stop. So when we get back, we'll talk about mutations and then viruses, and then again, I think we should be on track to have the exam on Friday.
What time will you be online?
Should be by 9. Hopefully I get on by 9, but.
Okay, I'm gonna go to the city guys. I need them all.
Perfect, okay.
Mr. D,